Welcome to the Pharmacy Inspection Podcast, where we discuss topics related to sterile and non-sterile compounding pharmacy in an effort to promote compliance and increase quality. The Pharmacy Inspection Podcast is a member of the Pharmacy Podcast Network, delivering quality and meaningful conversations and discussions about our pharmacy industry and the critical role pharmacists play in our healthcare systems. Learn more at pharmacypodcast.com. Please welcome your hosts, Brian Prince and Seth DePasquale.
Hello to all of our compounding friends and colleagues, and welcome to the Pharmacy Inspection Podcast, where we discuss all things compounding in an effort to further the cause for both quality and safety in the compounding industry. You are listening to the first and original podcast series in our industry, and we'll continue to push our educational and knowledge-sharing agenda. You know, it's hard to believe that it's already October of 2018, and we actually just celebrated our first full year of recording this podcast. Our original podcast was released on October 24th of 2017. So happy anniversary to all of our listeners. We're very proud to say that we've produced and released 32 podcast episodes in these past 12 months. And of course, we're not done yet. We've got a couple more episodes in the hopper for this year. So thank you to our audience. Thank you again for being part of our listenership, just giving us your feedback. What has really kept us going is your encouragement, seeing your emails and your responses and your questions come across through various social medias, and even getting to meet some of you face-to-face at conferences and hearing your words of encouragement. So moving into this next year, we will continue to have an open source philosophy of sharing the industry's best knowledge with you. Thank you for joining us again this week, and let's jump into this week's episode with our guest, Kim Kieffer. Kim, thank you for joining us again on the Pharmacy Inspection Podcast. You've been a guest for us in the past, and always you bring a wealth of knowledge. And about 2 weeks ago, I guess, I had the pleasure of seeing you speak in Oklahoma City at the ARL Quality Conference. And there was such a great presentation that you did. I was like, you know what, our audience has got to hear that and at least some of those things. So, but before we get into that, if you would please reintroduce yourself to our audience and tell us, a little about yourself, where you are now, how you got to this point.
Oh, thank you. Thank you, Brian. So I have worked in the compounding industry for about the last 17 years, 15 of which, or actually more than that, but it's hard to say something that's closer to 20. But the majority of that was working on the industry side, so working for the raw materials suppliers, doing clinical and technical support for compounding pharmacies, doing formulation support, physical chemistry support, calculation support, that type of thing. And in that time, I became, pretty interested in the various reference materials and also a lot of the unique problems that compounders face.
So, Kim, during your presentation in Oklahoma City, you had a lot to say, but if you would give us somewhat of a synopsis, like describe why the USP Drug monograph should be referenced when formulating from bulk active pharmaceutical ingredients.
Sure. So when they asked me to do this presentation, it was in response to a client that ARL was working, ARL being the sponsor. They were working with a client that had an out-of-spec result on some testing they had done. And Dr. Kupik called me and said, Kim, we're going through this and we just don't understand what's happening here. And he gave me the whole rundown of the situation. And I thought to myself, you know what? I need to consult the USP. I'm driving, so let me do that and call you back. And of course, when I got home, I was able to look into the monograph, because the monographs in the USP, which by the way supersede all the chapters. So what's in the monograph matters to your formulation, whether you're a drug manufacturer or compounder. So I was able to look and see, because in each monograph, there's different testing specifications, there's different acceptance criteria, there's different water contents, there's a different way in which the water contents are often applied. So you have to take that all into consideration when you're doing your math to start your formulation process. With compounding, we are not required under 795 and 797 to do a lot of end product or potency testing. For sterile products, there is some testing that's required if you want to exceed certain expiration era beyond use dates. But in terms of potency, we're really not required to do that. So what we have to do, or what we should do, is all of these sort of in-process checks along the way, and some easy ones to do are to make sure that you calculate correctly, that you weigh correctly, and that you do your variance calculations correctly. So those are just simple things you can do that will tell you, or at least give you some reasonable assurance that what you say you're putting in per volume or per unit is what it's going to be. But in order to do that math correctly, you need to make sure that you are taking into account all the specific variables of the chemical. And the only way to really see what those variables are is to look at the monograph. So there are 3 different kinds of monographs in the USP. There are bulk substances monographs. So those monographs describe what a bulk substance should be, like in terms of how much water it can contain, how much heavy metal it can contain, if there's a loss on drying, and that would be water and other liquid type substances that are in trace amounts left over, how active it can be, and on and on and on. And every one of them are a little bit different. And sometimes we want to make sure that we calculate off the water, because when you get your material in your bottle, it's often hydrated at least somewhat from the derivation process. And so that water, or maybe it's a salt form or an ester, those things actually dilute the activity of the base chemical. And for a lot of, in most cases, not all cases though, but in the majority of cases, we want to make sure that we are accounting for those things that are diluting the activity so that we can give the activity that we want. But like I said, in some instances, we want the diluted form. So it's really, and the only way to know that is to go in and look at the standards, which are contained in the monograph. So that's for bulk substances. But then we also have drug substance monographs or finished product monographs. And these are monographs for, most of the general things that we see, like lidocaine injection or ibuprofen tablets, or you name it, the stuff that's out there on the market, like there's a monograph for it. Now, those finished product monographs don't give you things like a formula, and they don't give you mixing instructions or beyond use dating, but they do give you the general backbone of the formulation. So like, this is in water for injection, it has a pH of X, it has an isotonicity of this, it has an osmolarity of this, it has endotoxin levels and that sort of thing. So we'll give you that backbone so you can sort of formulate from that. So that's the finished product monograph. And those are very important because we have acceptance ranges in there, and acceptance ranges for potency in particular. And that is something to keep in mind. We always think, 90 to 110%, that's what we use in compounding, and that's often a default in a lot of these. However, it's not the case for all of them. Just like for the drug monographs, we also have these potency ranges that are all over the place. They're not always 90 to 110%. So we want to look and see what the monographs tell us, because that's what we should be shooting for in our calculation. If it says in the finished product monograph and your compound is somewhat close to that finished monograph and that potency acceptance range is 95 to 105%, that's your range, not 90 to 110%. Because remember, the monographs always supersede the chapters, right? So even if somewhere else in one of our compounding specific chapters, it might reference that 90 to 110%, your monographs are really going to supersede that. So you want to make sure you look at it. And then we have a third kind of monograph, and that's our compounding product monograph. So USP has been busily adding compounded product monographs in so that we have good information on testing those products, on beyond use dates for those products, and mixing instructions, formulas, et cetera. So those compounded monographs actually have an acceptance range. They have a formula. They have mixing instructions, beyond use date information, and then all the analytical information to test that product, just like the other monographs. So those are there also. And again, if you're going to follow those formulas, obviously you would use the BUDs there, and you would, of course, use the acceptance range. So they give us a lot of information in those. So I think that is really, really important to always keep in mind. First of all, whatever 797 and 795 tell you, those monographs come first. So you have to look at those first. And you will never have all the information about doing a calculation unless you look. And a lot of times there can be some very significant water contributions that can push you out of your acceptance range, or in certain cases, there is a large water content that actually needs to be calculated in. So the only way to know is to reference. And it's so much fun to find all of the different variations. And of course, it gives you a better and more reasonable assurance that you're going to have what you want at the end of your formulation and production process if you have good math in the beginning.
So finish the story where Dr. Kupiec called you and wanted more information, and you, of course, had to consult. So what was the actual situation there? So give us that real life example.
So it was a atropine sulfate formulation, and I unfortunately don't remember if it was an eye drop or an injection. But the issue is that they were looking for an acceptance range that was specific to that, and they were also looking for an activity range that was not adding up. And it was because of the way atropine sulfate is described and how the water content is calculated. So they did not account for the water content appropriately. And had they looked at the monograph, that would have taken care of their issue. And what's interesting about atropine sulfate is it's one of those things where the water content is calculated in, and also instead of taken out. Also, you're keeping the sulfate, even though the bulk substance monograph eliminates the water, the finished product keeps the water, and you're also not removing the salt form. And then in addition, depending on what you're making, whether it's a solution for injection or eye drops, or it's a tablet or an ointment or whatnot, there's about 4 different drug monographs or finished product monographs I can think of. They all have different acceptance range in terms of what's acceptable for potency.
Seth, would you like to interject at this point? Because I have nothing else to share.
Yeah, give me 1 second. Just connecting.
Kim, you actually used that act as an example in yours. So like slide 34 to 47.
Mm-hmm.
And water MW. What is MW? What does that stand for? Water MW.
Let me find it. Molecular weight.
Molecular weight. As molecular weight, you say you did a formulation of 18.01 divided by 694.8, and basically came out to 2.6%. Is that worthy of?
I was just demonstrating there that, so for this one, I was really just demonstrating there that if you were to go in and take the water out using the molecular weight, because the molecule is a hydrate, so the molecule is displayed and listed with the water in it. And so if you just do a base or a molecular or a salt, base salt conversion where you just pull off the salt and the water, or you just pull off the water only and not the salt, that accounts for like 2 point whatever percent. Now, if I go in and I actually use the C of A and look at the actual water content, because there's a range for the water content, it might be a slightly different range, but it's going to be really close to that 2.6 or 2 point whatever it was. So that was just like a side demonstration, because the USP gives you a way to account for hydrates and salts by using the molecular weight. So instead of looking at the C of A, I think what I was conveying in the lecture is that, you need to use your C of A and apply those specific values, because that's going to get you closer to your base activity. But you can do it just by taking off the molecule. Like, here's the molecule, it weighs X. If I take off these parts of it, which weigh X, then I get this. And that's the way USP shows you to do it. But you can get a lot closer and more specific if you use the actual values from your C of A. So it's kind of like you have to use your C of A, but you have to look at the monograph too. So I was just demonstrating that as like an aside. Like if you don't believe it this way, well, look, it's right here. I don't know if it was really relevant, but I thought it was interesting at the time when I wrote the slide.
I like how in your presentation you're using a lot of examples that are like very narrow therapeutic index drugs, like theophylline, levothyroxine, liothyronine I see on here. So I feel like pharmacists have a good appreciation for, okay, I know that this is a narrow therapeutic index drug. I should probably look at and see what I'm doing and concentrate on this. Because if I screw this up, this could be very detrimental. So, is there like outside of the monographs, what specific chapter would you point to that you would kind of tell pharmacists or techs to check this out, make sure you're on point at the very least with your narrow therapeutic index drugs. But it would also give them an idea of like maybe what they should be looking at across the board for all their other drugs and compounds.
So unfortunately, you can't. I picked examples that were near and dear to people's hearts, like the levothyroxine, liothyronine. Those are problematic in a few ways because the liothyronine is given as the base and not as the sodium. So there's two parts. You have to worry about the base salt conversion and if there's water content and if there's an appreciable difference in the potency, that you have to account for. And then the levothyroxine, which so often we give at the same time, is given as the salt and not the base. And it has a ton of water content usually that has to come off. Plus you have to adjust for so there's that. And then on top of it, you have to make an aliquot. And then, and so there's all of these problems that arise. And we know that people have problems with those. We know that people are not comfortable with those calculations. We know that they fail when they're tested a lot. I mean, I know this because I have helped just, hundreds and hundreds of people with those questions. So I use those specifically. But when you get into other things, like I also use benzoyl peroxide because that's not narrow therapeutic index, but it has a ton of water in it. And if you didn't know that because you didn't look at your C of A or the monograph to see how it's given, you could seriously underdose it because it's going to have at least 20% water in it no matter what, because otherwise it'll explode. So that's another indication. So I think I tried to pick a handful of things that were just really odd because you never know what the monograph is going to say. So if you don't already know what the monograph says, you have to look at it. So I would say anytime you have a new drug or a new formulation that you're preparing and you haven't really reviewed that monograph, you should probably see just in case there's something funky. Because like in the case of atropine and in the case of morphine, where it's a pentahydrate, you give it as the pentahydrate. If you calculated that water off, you could, overdose your patient possibly. So, I mean, you don't really know until you look at it. And we get into this, well, morphine sulfate is morphine sulfate, and that's what we call it, but it's always a pentahydrate. But then there's lidocaine hydrochloride that's almost always a monohydrate, but we calculate the monohydrate off. So there's just always a different little caveats, and it's not always with narrow therapeutic index things. But aside from the monographs, USP 795 and like the first or second paragraph gives us a list of chapters that are really important, and they say you should be familiar with these. So there is 1151, which is a pharmaceutical dosage forms, and I think that one's really entertaining. You probably could get by without reading it, but it gives you really specific information about what the definition of a dosage form and what constitutes an extended release versus a control release capsule and that sort of thing. There's also 1160, which is pharmaceutical calculations. And I don't really think that there is a single compounder out there that never has a calculation question. So there's a lot of really good information in there. There's the quality assurance chapter, which is 1163, and that gives information about doing variance calculations, so those are really weight variance calculations, and there's specific information about doing it for dry dosage forms and for like ointments and creams and things of that nature. And if you're not doing any sort of analytical laboratory testing, those weight variance calculations can be really, really helpful for doing a little QC of your own products. There's also there's a whole chapter on labeling, which has kind of come into focus recently. There was a MedWatch letter that FDA shared with everybody a couple weeks ago that specifically called out several outsourcing facilities. I know it's outsourcing facilities, but 503A compounders do this same sort of stuff notoriously. It's basically, they were saying the packages were not labeled correctly because there is specific guidelines, and they come from USP, of how to label injectable products in terms of like the strength per volume of container, and then later on in parentheses the strength per ml. So the industry standard and what's in Chapter 7 of USP is how to do that, and compounders haven't traditionally done it that way. They either do it backwards or they don't give both information, whatever. But in this letter that FDA shared from MedWatch, it essentially showed the packaging. It told who the manufacturer was or the outsourcing facility was. So we were specifically called out on that. And the worry is because we have injectable products labeled one way in industry, and then these get into the market, people will be confused because they're used to seeing them the other way. And I think it's really relevant because I think as compounders, we feel like we live in this bubble where we can just do things how we do things, but there's this whole world of manufacturing that is now closing in on us, reminding us that we are drug manufacturers, but we have some exemptions under 503A and 503B, whichever one we're operating under. But labeling is not something that we have an exemption from. I mean, we do have to look at that and see if we probably should incorporate that into our practice. And there's a bunch of others. I mean, if you have a question on anything, you can probably find a chapter that can give you some information on that. But those are the basic, there's like a list of them, the basic ones that you should know about as a compounder.
Yeah, I think 795, does 795 mention all the ones that you just mentioned? Or
yes, it does. It's okay. Like in the first couple paragraphs of the chapter, it says, here are all of the other things you should know about. And it gives a handful. And there's about 10 of them.
Nice. Well, you and I offline, I think, have had the same conversation. It's crazy helpful. It really got me into looking more at the other chapters within the compendium, and keeping the compendium handy, basically on my phone. So
yeah, it's nice, right?
Yeah, it's really nice, actually. Brian, do you have any other questions for Kim? I mean, honestly, this has been super helpful, and Kim is such a wealth of knowledge when it comes to this stuff. So,
yeah, no additional questions. I was telling Kim actually that on the way to dinner after she spoke, then we as a group went to dinner later, and there was still a lot of conversation about the things that she brought up, and it's interesting at what we think we know we don't know, right? That's kind of our favorite statement, is that we don't know what we don't know. And I'm sure that this could go further and further, and I'd love to talk about it, but of course we're coming up on that timeline. So Kim, we'll give you this opportunity. Any last pieces of advice or anything you just want to throw out there to our audience before you part ways?
Just that if you don't have the USP and it's not something that you use as a regular tool, I think people use Tristles, and maybe they use Martindale's and possibly the Merck Index, or they can just do Google searches or whatever. But like, if it's not something that you're regularly using as a reference tool, I strongly recommend you get it, and that you get the full compendium and not just the compounding compendium, because it does not contain all of the monographs. And just start using it, because there is so much information, and I think we've lived in this, like I mentioned before, this bubble of compounding where we just do what compounders do, but we really are drug manufacturers on whatever scale, exercising our exemptions. But the USP is dedicated to, just as much dedicated to compounding as it is to pharmaceutical manufacturing or commercial manufacturing. So they're really working hard to supply good standards and good information, and to not use it is just passing up just a really great, awesome tool that can really answer a lot of questions. So get it if you don't have it.
Excellent. Excellent.
So really great advice, Kim. We as always, we appreciate you're welcome on this show anytime, because you certainly bring a lot of great pieces of information that we need to know. So thank you again for joining us. You're always welcome here, and take care. I completely blew that outro, didn't I?
No worries.
Wow.
Clean that up.
Yeah, I just had a long day. I've had Monday all day, and I'm just smoked and done. So anyways, thank you, Kim, as always.
Sure. Well, thank you. Fun.